But as we advance as a field, you really quickly see that that is not the biological situation whatsoever. And if you want to go more biomimetic, if you want these cells to do things over time, divide, and then differentiate, you need different environments. And so we really have a challenge in front of us now as a field that we have to make these matrices that can adapt and can change over time to the cells. And in fact, we started with just a simple version where our matrix is constantly dynamically adapting, and we find already major benefits.
For decades, we've grown cells flat on plastic and called it biology. But cells don't live in 2 dimensions. They live in a soft, responsive world that pushes back. What if the gel surrounding a cell wasn't a passive bystander, but an active conversation partner? Today, I'm joined by Jan Hunik, CEO of MosaMatrix, and Matt Baker, its CTO, and the inventor of a new class of hydrogels. Let's build the cells world together. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Jan and Matt. It's good to have you on today.
Thanks. It's great to be here.
Thanks, David, for the invitation. Great opportunity.
A pleasure. Let's start with this perhaps controversial question. Share something that you believe about bioprocess development that most people disagree with.
I think the mechanical biological behavior of hydrogels is really a crucial aspect of natural physiology and morphology behavior of natural cells.
David, this is a tough question, but I think this is always a good one to start with. I think a lot of people would disagree that materials are critical for the future of tissue engineering. I think a lot of people think that you can do this with cells alone, and I would strongly disagree. And not just from the materials point of view, but also from the process point of view. I think it's going to be critical as we scale up, but we have to get these materials right.
Tell us about yourself before we move into the science. What sparked your interest in the field and what eventually led you to founding MosaMatrix? And what were some perhaps interesting pit stops along the way?
Okay, this is an interesting one. I'm a scientist. I find it really interesting that if we can make and we can control things, I think Feynman nicely said, what you can't create, you can't understand and control. And for better or worse, sometimes a believer in that philosophy. But I also had always a really big interest in how things made it to the market and the fact what dies in the lab becomes useless to society. I think my biggest interest started when I took a night class at Clemson University during my undergrad, and I had a CEO of a company come and teach us how to commercialize things. And saw it was totally different than the lab. And just being able to think that way really inspired me. We always wanted in our research group to start to try to commercialize some of our technologies. And we found that some of the base technologies from MosaMatrix was a good shot to start with. And I'm also a firm believer that you at some point just have to get started and see how it goes.
Yeah, excellent. How about you, Jan?
It really started for some microbiology lessons, microbial physiology at university a long time ago. And then you can see how versatile biology is. It's amazing what you can do with it. Look around you. I mean, it's all biology if you can use it at the best of your own benefit. I mean, it has been a remarkable journey for close to 40 years, and it still is. And I'm really intrigued by how that technology has developed to the point where we now can grow human cells. And maybe as a MosaMatrix, we can really give that opportunity a big boost.
Matt, take us back to the beginning. What was the problem you encountered that convinced you the field needed a generally new class of hydrogels?
This one's actually quite simple. I worked in an area of chemistry called supramolecular assembly, where we worked on dynamic molecules flying around and making these superstructures. And we realized that, in fact, that's what your body's really good at. Your body's really good at building up these matrices from smaller protein fragments, having them respond to stimuli, change properties over time. And we looked at current hydrogels and saw that, in fact, we weren't really good at doing that. And our group was trying to see if we could take this fundamental way of making matter and bring that towards hydrogels quickly for tissue engineering and just try to make things more dynamic, a little bit more lifelike, and see how powerful that was for the field.
And you, Jan, when you encountered Matt and particularly his hydrogel work, what made you believe that this could be a company worth building?
I was really intrigued by the beauty of the simplicity. One large molecule and two smaller molecules. You can make a world of different conditions for those cells. But from a business perspective, I also saw the regulatory hurdles to get it to the market are extremely low. And of course, I'm looking at a market that is fast moving. So there's new developments can quickly be adopted by this market. So without looking really into all the details, that was really the starting point. And we will come to those details later, I think.
So Matt, tell us about the hydrogel, because not every smart biotech scientist listening might be familiar with that. Usually what comes into my mind is a passive scaffold cells just sit in, but why is that picture wrong, and how does actually your matrix work?
I won't say that picture is wrong, because in fact, that's what a lot of the hydrogels out there currently do. The scaffold challenge is fun for bioengineering because at first you really need something that is a bit static, so that way you can make it reproducible, you can get cells to grow into it. But as we advance as a field, you really quickly see that that is not the biological situation whatsoever. And if you want to go more biomimetic, if you want these cells to do things over time, divide, and then differentiate, you need different environments. And so we really have a challenge in front of us now as a field that we have to make these matrices that can adapt and can change over time to the cells. And in fact, we started with just a simple version where our matrix is constantly dynamically adapting, and we find already major benefits. Cells spread differently. They're able to move the matrix out of the way and create their own tissue, all things that you can't do in these more static hydrogels of the first generation, if you will, tech.
How do they adapt to the cell? How does that work?
They are dynamically reconfiguring. The bonds are moving around in real time. So they have the ability to, if a cell wants to produce their own matrix, for example, they can kind of push against the material. The material will give it that space. But also as they start to pull on the material, they can start to reconfigure the material and create space for themselves as well. It's something that we're actually trying to get better at characterizing on the academic level, but on the product level, it just works.
And what are the important characteristics of the gel? What comes to my mind is like stiffness or how it relaxes, especially as you're talking about more dynamic one. Tell us a bit more about that.
The hydrogel stiffness is really critical to getting cells to do what you want to, especially as you're trying to create tissue or go towards more bioprocessing. And I think that was shown maybe 20, 25 years ago in a few landmark papers by Engler and Disher. But recently, in the past 10 years, we've seen that how a gel relaxes these stresses is also critical. That's where you get into the biomimetic aspect. So biological tissues relax stress very well because they're large molecules that can move past one another. And we weren't so good at doing that synthetically until just recently. And we are one of the first hydrogels on the market that has tunable stiffness and stress relaxation. So we can really start to show that we can get good tissue formation and we can tune it based on the customer's need.
Now, looking at the company evolution, Jan, you started with the ambition of 3D printing human tissue. Your focus has now changed. Tell us a bit more why you started out with that and what drove that pivot.
I think it was a good start to think about that part on 3D bioprinting. But what immediately attracted my attention was that the conditions for the cell growth were also optimal. So Matt showed in some articles and publications that the morphology and the physiological characteristics of the cells were also much better. And then last year, I was on a trained mission of the Dutch Ministry of Economic Affairs in Switzerland, listening to stories of pharma companies and startup companies. And then I started realizing that one of the problems that industry is facing is how can we make not only have that matrix and hydrogels for those cells, but also to use them in high-throughput screening systems. It's really a hassle at this moment, and it's really hindering that application for the drug discovery market. And then when you think about 3D printing, for 3D printing, you need to have a very well-processable hydrogel. Now, one of the things that you have in the 3D printer is that you need to mix, to pump it around, to put those droplets in a specific spot. that would ideally fit in high-throughput screening systems. So with that, there's two elements combined. We very fast came to the conclusion that it really makes sense to focus ourselves on 3D cell culturing for drug discovery with the application of high-throughput screening systems. And since then, we see a lot of traction or approached by companies that want to work and try our material after they have heard about it.
What is your target these days? What kind of application or people would benefit from this technology versus using older systems?
There are several important reasons why people can use this, but we are a very versatile platform. So we are not bound to one particular cell type, especially because of the adaptation that Matt explained, that we can tune those properties very well. There's a wealth of type of cells we can accommodate. And on the other hand, and I think that's also very beneficial in this industry, is our material is much more reproducible than what's currently out in the market. And that reproducibility is something that really hinders this application in the drug discovery field. And there we have a big advantage. We should benefit from that.
And how does this advantage of the new technology, what I'm hearing, it's better controlled, it's more suited to what cells need. What does actually change in the quality and reliability of the results?
There's something that we have to show. For the moment, what we see is the current materials are really a frustration, especially people in that research field, and also try to find applications. And our material is not animal-derived. We can make it in a very pure form and well-described chemistry. I think from that point of view, we should be able to make it much more reproducible, or we already make it much more reproducible than what's out in the market at this stage.
As you're further developing this technology, I'd be curious about what is your strategy to get early customer feedback, to really understand what your target audience needs. What is your approach there?
We work with academia to learn more about, and that's also a very useful pathway to learn more about how our material behaves with different cell lines, and we are building that information. But to give you an idea, we recently entered two large consortia. One consortium is looking at how can we make better 3D cell culture, bring that to the marketplace as being a valuable tool. That's a large consortium of 50 companies that work together. We are one of the suppliers of the matrix material for those living cells. And since a week, that's now official, we are also part of a consortium for improvement of kidney dialysis by using living human cells. And that consortium is funded by the Dutch Patient Organization for Kidney Patients. That's also a consortium with academia and companies working together to move this technology forward and see if we can improve really patient lives there with our technology. I think that very well describes the different fields where we try to find applications. So directly into the business, working with potential customers together. And secondly, working with academia, preferably in consortia, where we bring in the matrix. Other people bring in their cell technology and try to move this forward to really applicable end product into the market and to patients.
So you work in direct collaboration with various partners to get instant feedback, shall I say?
Yes, quite interesting, sometimes very confusing. So then we need a lot of discussion with them.
I imagine.
But we're moving forward, definitely.
I'm curious if you can share it. What were some interesting feedback you got, or perhaps ideas you would not have thought of?
One of the things that we encounter, for example, is that the media are very diverse being used. And I think the impact of the media is much higher than we think. And also the cells are influencing the way the matrix will behave mechanically and also in the application. So there is it's biology. So all those interactions need to be taken into account and understand. So it will be a journey, but the initial results are looking very promising.
That's fantastic. I'd love to dive into the media since you mentioned it. And I imagine, and especially within your field of application, a lot of people are still using animal-derived components or perhaps non-defined components. What are the challenges there? Where are you at today with this kind of development?
Our material is not animal-derived. I think that's also the main reason why we can be so much more well-defined than what's out there. That helps. But we know that there's a lot of animal-derived materials around, and those are a mix of not only matrix, but also signaling molecules, growth factors, whatever you can imagine. As soon as they're out, what's left over, of course, is the interaction between our material and the cells, and of course the media. And that's something that we have to further understand and work around it and make sure that our material performs what it should do. And by the way, we can also functionalize our material, but that's something that Matt can tell a little bit more about it to make it more suitable for different applications.
I think it's a nice question to think about because, in fact, mammalian cells were not evolved to grow on plant-based materials or non-animal-based materials. So scientifically, you have a giant challenge there. I think that's where the science and the technology become really interesting. How do you put the pieces back in that are important for cells? And then what it enables for the field is all of a sudden, if you don't rely on animal matrices anymore, your ability to scale and your ability to open up the processing window becomes enormous. So I think it's a really important transition for the future of the field, not just from an ethical point of view, but more from really opening up the technology point of view. But comes with some big scientific hurdles. You needed to make something biocompatible where evolution did not evolve in that direction.
Yeah, this is definitely a challenge for a lot of cell types, no doubt about that. And speaking about the challenges, since you're using all kinds of different cells combined with your hydrogel, how does the scaling work? I don't know if you can talk about scale up, or should we say rather scale out? What are some technical challenges people face with this kind of cultures?
I think there's a lot of challenges. One thing that's really in front of the field right now, reproducibility is always a challenge, so I'm not going to talk about that too much. That's in front of us all. But two big challenges for this field specifically is the characterization. So once you make something in 3D, it takes a much longer time to characterize it, and then you can't measure and make sure that your process is working well. And then on the other hand is the technology to scale. So both the instrumentation or the machining, but then also the materials that allow this scalability while keeping cells alive. It's not a problem that science has really tackled in this field in the past so well, or with so much vigor, let's say. And so that technology push is coming along, but then there is this challenge of being able to actually measure and know that what you made is good, how to make these changes on the fly.
And how does this impact the way you think about quality control, for instance, for a process?
As a chemist, we have decent quality control standards in some of the measurements that we make from the chemical point of view. So we're relatively confident that our molecules are what they are, and we can ensure that. As you start working with customers on these much more complex situations where we're part of a high-throughput platform now, or we're helping to create more organoids along the way, it makes you think about it quickly. Let's say it that way, where that becomes a conversation that we have with customers very quick. How do we measure success here? How do we know we're going in the right direction? Also to make sure that our culture of trying to have good quality control matches with the customer's ability to also measure and make sure that what they're doing is they have confidence in.
And what is key in order to be able these organoids? Because they're difficult to standardize and to scale. How can you run them at higher throughput and, shall I say, higher scale?
Jan wants to answer that one. We'll let him go.
The interesting thing I see is two things, observing when I was talking to some external parties that we work with. First of all, they want to make it smaller. So they want to have very small cultures. They go from 96 well plates to 84 well plates. And all the thing that they see is by automation, they improve the reproducibility by using automation, if they can, because that's their major struggle. Automation has two functions. One, can we have a higher throughput, but also can we improve the quality and the consistency of the data? I think that's one of the reasons why we can become an attractive partner. We can benefit both. And that's also what I hear back from them. Let us show you data, because we need to generate data. If that data is good, they will give this a try and see what they can do with it. Then small scale means that you can just screen more cultures in a shorter period of time.
The scaffold was never passive. Stiffness, stress, relaxation, the whole mechanical handshake shapes the biology long before any drug enters the picture. We've traced how a chemistry problem became a company and why getting the matrix wrong quietly corrupts everything downstream. In part 2, we head into organoids, throughput, and quality control. If this reframed how you think about 3D cell culture, leave a review on Apple Podcasts or your favorite platform. And thank you so much for tuning in, and we'll see you next time.
All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.